Namaskar. Now I am going to discuss the two different pathway of P53 protein. The tumor separation gene P53 produce the P53 protein is very important in regulating cell cycle during the G1 and S phase. During the G1 and S phase progression. So first pathway, it is a very important protein which sends the DNA breakage, DNA damage. If DNA damage occur in the G1 phase or during the G1S phase, what happened? DNA damage by any kind of stress, either UV ray or any kind of ionizing radiations, non-ionizing radiations, whatever may be, DNA damage is immediately sensed by protein ATM-ATR. This is a very important protein which sends the DNA damage. When DNA damage occur, this ATM and ATR sends the activate P53 protein. P53 when is normal condition. Each and every pathway. First, I am very clearly you have to understand that every pathway has two parameters. One, that when all the genes are okay, and another is when any one of the gene is getting mutated. Now it is here all the genes are okay. That is what will happen if DNA damage occur under such condition. All the genes are okay and DNA damage occur, then ATR sends the DNA damage. First, it is first sensed by or first observed or identified by the ATM and ATR. Then immediately ATM and ATR kinase protein, they are phosphorylating. This protein phosphorylating the P53 gene, and P53 is getting phosphorylated and activated. After activation of P53 protein, it activates the P21 gene, P21 protein. P21 protein when getting activated, it's a direct inhibitor. In the previous video I have discussed about P53 function, that is P53 directly inhibit CDK2, cyclin E, CDK4, 6 protein. That is all CDK proteins present here. That is G1 and G1S phase. The CDK protein and cyclin CDK complexes are directly inhibited by the P21 protein. So what happened? DNA damage occurred, ATM-ATR getting sensed, and they phosphorylate P53 gene, activated. P53 gene is activated here. P53 activated, it activates the P21. When P21 is activated, it directly inhibits the G1S and G1 CDK cyclin complexes. When G1 and G1 CDK cyclin being deactivated or inhibited, then immediately stop the cell cycle. How? When they are inhibited, these are actually kinase protein, cyclin dependent kinase protein. Their kinase activity will be hindered. When kinase activity hindered, CDK2, cyclin E, CDK4, 6, then they are not able to phosphorylate. They are not able to phosphorylate the RB gene, the retinoblastoma gene, another tumor suppressor gene. And this RB gene is very much important because RB gene is getting attached. RB gene normally under normal condition, under non-unphosphorylated condition, RB gene is attached to it E2F transcription factor protein. E2F transcription factor protein is attached to it RB under non-phosphorylated condition. So when DNA damage occurs, it is non-functional, and under this condition RB is not phosphorylated because the RB phosphorylation is totally dependent upon the activity of CDK2 and cyclin E complexes. So RB when non-phosphorylated, it is attached and inactive condition, remain inactive condition with the E2F protein. So under this condition, E2F protein is not able to transfer from cytoplasm to the nucleus. So it is not transferred to the nucleus. As a result, if E2F is not able to transfer into the nucleus, it will not transcribe the other cell cycle proteins responsible for the transfer of the cycle from G1 to G2 phase. So E2F is arrested here. As E2F is arrested here by RB, then cell cycle progression is stopped because E2F is a transcription factor and it is not able to transfer to the nucleus. So the the transcription of several important genes responsible for the movement of the cell cycle from G1 to S phase is not able to transcribe, and cell cycle arrest. Cell cycle arrest is G1S phase. This is the normal phenomenon, that is, if DNA damage occur, ATM activated, P53 activated, P21 activated, inhibit. P21 when activated, inhibit the CDK2 cyclin complex. When it is inactivated, RB is not phosphorylated. RB is not phosphorylated, then E2F is not functional. E2F is not able to transfer from cytoplasm to nucleus, and as a result, all the transcription of the important genes that are responsible for transfer a cell from G1 to S phase is not able to transcribe, and as a result, arrest of the cell cycle occur at the G1S phase. Now, this is a common pathway. Now, if DNA damage not occurred, DNA damage is not occurred, normal DNA, then what will happen? ATM ATR is not getting sensed, and P53 will be not activated, and P21 is in activated condition, and CDK2 cyclin, these are present in active condition. When they are present in an active condition, what will happen? The RB will be phosphorylated. Then RB will be phosphorylated. When RB will be phosphorylated, then E2F will be active. E2 will be transferred from cytoplasm to nucleus, and all the genes responsible for the transfer from G1 to S phase will be transcribed by the E2A protein. So no arrest of G1 to S phase cell cycle. Now this is a common pathway. If any gene will be mutated in this pathway, that is the P53 or ATM or ATR, any gene will be mutated, then what will happen? If they will mutated, they will be not able to sense the DNA damage. Then what will happen? DNA remain in damaged condition inside the cell, and no signal is transferred to the CDK2 cyclin. CDK2 cycle will continuously transfer the cell or divide the cell in damaged DNA condition, with damaged DNA condition. In that condition, cell cycle will progress continuously with defective DNA, and cell will be cancerous. This is. And another pathway, the same pathway present here, in a DNA damage occur, then ATM ATR is activated, and another cell cycle checkpoint protein is there, CHK1. This will be activated. When this will be activated, this will directly inhibit the CDC25 protein. This CDC25 protein is a phosphatase, and these proteins has immense ability to activate the CDK cyclin complex. Now, when DNA damage occur, then cell have to stop the cell cycle. So what happened? Here ATM is activated, CHK activated, and CHK when activated directly inhibit the C25, because if it will not be activated, then CDC25 will activate the CDK2 cyclin complex. When this will be activated, then cell cycle will continue. As you know that this will be activated, CDK2 cyclin is activated, cell cycle will continue. So in this way, it is regulating the cell cycle under stress condition. Now, another important here, the P53 function is that under several cervical cancer is responsible for more than 50% cases. Cervical cancers are due to P53 mutations. Why? Here, papillomavirus, you know that the human papillomavirus, HPV, human papillomavirus, are responsible for the cervical cancer. Human papillomavirus is a DNA virus, and if infect the epithelial cell of a human being, mainly females, then what happens? They are present in extracellular plasmid in a epithelial cells nucleus. Extracellular plasmid. Then extracellular plasmids, and accidentally if they are present in a integrated inside the host genome, that is, if viral DNA will be integrated into the host genome, then immediately they will produce, the virus genome will produce huge amount of E6 and E7 protein. Then E6 and E7 protein are very important. E6 protein directly inhibit the P53 function, and E7 protein directly inhibit the retinoblastoma gene, RB gene. As I already mentioned that what is actually the function of P53, as well as when P53 is inactivated, then P20 will not be activated, will be inhibited. This will not in inactive condition, this will present in inactive condition. When this one is present in inactive condition, CDK4 will be active. When CDK4 is continuously active, then it will phosphorylase the RB gene. When RB is activated, then E2 is free, E2F is free to transfer the G1S continuously. So G1S will not be arrested, cell will not be arrested at the G1S cycle, G1S stage. So this will form a wart or tumor. So in this way, papilloma virus produce the E6 protein in females, cervical cancer case, and E6 protein inhibit P53, P53 inhibit P21. When inactivated, CDK4 cycle, CDK4 will be activated and cell cycle will continue. Now, E7 protein, how they will function? E7 protein inhibit directly the retinoblastoma gene. As in that retinoblastoma when inactivated, directly inactivated, that is, it is not phosphorylated. When and then what will happen? Directly inactivated, that is RB gene is it inactivated by phosphorylation. Here RB gene getting phosphorylated. When RB phosphorylated, E2F will be free, and E2F when E2F will be free, then this is a transcription factor, cell cycle will continue. That is, it is very important that E6 and E7 protein directly activate the G1S cell cycle regulation point. So this is very important. Human papillomavirus, after entry, they will produce E6, E7 protein. E6 protein, what happened? E6 protein directly inhibit the P53, and P53 inactivated, P22 inactivated. This is the common cycle. And in case of E7, this occurs in the same way. That is, RB is inhibited by phosphorylation and E2F is free. So cell division continue.